Welcome, everyone. It's a great pleasure to open the webinar Zebrafish as Experimental Model for Research. I'm Rafael, and I'm Associate Professor at São Paulo State University. And I would like to present my co-chairs of this webinar, Dr. Ivis and Dr. Natalia. Welcome.
Hello, everyone. Thank you for joining us to webinar. My name is Ives Charlie. I am a researcher at the University of São Paulo.
Good morning, everyone. My name is Natalia Feitosa, and I am an associate professor at Federal University of Rio de Janeiro. First of all, we would like to thank the 1,200 subscribers from the different parts of the world that are connected with us in this webinar. Thank you very much for making our event possible. I also take this opportunity to thank our staff team who worked really hard to make all the things well organized for this webinar. Mayra, Isabella, Larissa, Felipe, Carol, Renata, Bianca, and Luis Henrique. I would like to thank as well, special thanks are also given to the technical staff from AGDC, Institute of Bioscience, UNESP, Institute of Biomedical Sciences, USP, Federal University of Rio de Janeiro, Oswaldo Cruz Foundation, and our sponsors.
So now, before we start, I would like to invite everyone to watch a video from the 4 institutes that supported our event.
Saúde, direito de todos e dever do Estado. Direito à moradia, ao emprego, à educação, a um meio ambiente saudável, à água potável, ao lazer, à cultura. Direito à voz e à paz. Respeito à diversidade. Saúde para a Fundação Oswaldo Cruz abriga todas essas dimensões. É assim desde 1900. Uma visão integrada da saúde. Na pesquisa, ensino, assistência, informação, comunicação, memória, na promoção da saúde, na inovação, onde permanentemente se pensam respostas para as necessidades da sociedade, onde se faz ciência em defesa da vida. Você pode até não saber, mas carrega a FIOCRUZ dentro de você. Desenvolvemos e fabricamos vacinas, medicamentos, biofármacos, testes para diagnóstico. Formamos pessoas do nível médio à pós-graduação. Realizamos pesquisas para superar ameaças à saúde, para diminuir riscos ambientais, para prevenir doenças e agravos. Pesquisas que beneficiam crianças, adultos, idosos. Uma instituição pública e estratégica de Estado, integrante do Sistema Único de Saúde, com uma rica história de contribuições à sociedade. Presente de ponta a ponta no Brasil, onde cada trabalhador é um elo forte e ativo. Nela, ciência e saúde cumprem uma função social para o país e o mundo. Pés fincados na tradição, olhos voltados para o futuro. Somos patrimônio da ciência e da saúde, da humanidade, do povo brasileiro.
O Instituto Nacional de Controle de Qualidade em Saúde, INCQS, foi criado em 1981. É uma unidade técnico-científica da FIOCRUZ que atua em áreas de ensino, pesquisa e desenvolvimento de tecnologias de laboratório relativas ao controle da qualidade de insumos, produtos, ambientes e serviços. Parte integrante do SUS, é elemento do Sistema Nacional de Vigilância Sanitária, atuando em estreita relação com a Agência Nacional do Setor, a Anvisa, laboratórios de saúde pública, vigilâncias sanitárias estaduais e municipais, entre outros entes. O INCQS é o único que realiza ensaios em lotes de sangue e hemoderivados utilizados no Brasil e é a instituição responsável pela análise laboratorial para a liberação de lotes de vacinas e de soros hiperimunes produzidos ou consumidos no país ou para exportação. O Instituto também avalia reagentes para diagnósticos, conjuntos, artigos e insumos para a saúde e para a diálise, medicamentos, cosméticos, saneantes e alimentos, por exemplo, detecção de níveis de agrotóxicos de drogas veterinárias e de transgênicos, e analisa amostras relacionadas à saúde ambiental. Além da atividade laboratorial, o Instituto emite pareceres sobre questões técnico-científicas relativas à vigilância sanitária. O INCQS é acreditado pelo Inmetro em diversos ensaios laboratoriais e serviços de calibração, tendo um sistema de gestão da qualidade consolidado e eficiente. Inclusive, é pré-qualificado pela Organização Mundial de Saúde nas áreas de medicamentos e vacinas. Este é o Instituto Nacional de Controle de Qualidade em Saúde, na FIOCRUZ, contribuindo para fortalecer o SUS em benefício da população brasileira.
O Instituto de Ciências Biomédicas da Universidade de São Paulo é uma referência nacional e internacional de qualidade no ensino, na pesquisa e nas atividades de cultura e extensão. Fundado em 1969, o ICB está localizado na cidade universitária, em São Paulo, com instalações em 8 prédios. Possui ainda uma unidade na cidade de Montenegro, em Rondônia, e um posto avançado em Cruzeiro do Sul, no Acre.
O ICB está estruturado em 7 departamentos: anatomia, biologia celular e do desenvolvimento, farmacologia, fisiologia e biofísica, imunologia, microbiologia e parasitologia, os quais contemplam as principais áreas das ciências biomédicas. Nosso ambiente é multicultural e amplamente democrático. Aqui no ICB, valorizamos a conduta ética, respeitamos a diversidade, incentivamos a consciência crítica e capacidade criativa dos nossos alunos, funcionários e professores.
Em meio a esse universo multidisciplinar, o ICB completa seus 50 anos com uma excelência consolidada e busca formar cada vez mais profissionais que produzam conhecimento e inovação, de modo a contribuir para o desenvolvimento da nossa sociedade. Seja você também parte do nosso Instituto.
Somos o Instituto de Biociências de Botucatu. Somos 950 alunos de graduação, 400 estudantes de pós-graduação, 150 técnicos administrativos e 130 docentes. Juntos somos uma grande aglomeração. Aglomeração de trabalho, de pesquisa, de ensino, uma gigantesca aglomeração de conhecimento. E este ano nosso IBB faz mais um aniversário. Já são 57 anos de história. Mas esse aniversário é, sem dúvidas, não da forma como eu e você imaginávamos. Pois eu te pergunto, onde está todo mundo? Em meio a essa pandemia, fomos todos surpreendidos e de uma hora para outra transportados aqui para esse mundo virtual. Um mundo que a gente conhecia apenas como entretenimento, mas não era um mundo real para as nossas aulas, as nossas pesquisas e atividades de extensão. Até porque estávamos acostumados a conviver fisicamente, com os corredores, os departamentos, os laboratórios sempre cheios. E aí, fomos todos virtualizados e nada mais era como a gente conhecia. Dá a impressão que perdemos a nossa identidade. Mas, na verdade, nós fomos desafiados. E embora tenha sido muito difícil no começo, percebemos que temos uma imensa capacidade de mudar, pois criamos novas conexões e novas maneiras de nos comunicar. Aprendemos a aprender e vimos que somos resilientes. Cada um com o seu conhecimento, com sua dedicação, não mediu esforços para, num trabalho único, reconstruir o IBB que permaneceu conhecesse além da sua estrutura física. Nós nos reinventamos. E nesse aniversário tão diferente, eu sei onde está todo mundo. Talvez nunca estivemos tão próximos. E com certeza, e que não importa qualquer outro desafio que nos espera, nós estaremos sempre aqui no IBB. Parabéns a todos nós.
Nos dedicamos a ensinar, a inovar e a transformar por meio da ciência e da atuação social responsável. Essa é a missão, há 45 anos, da UNESP, a Universidade Estadual Paulista. Uma jovem instituição, com 34 unidades em 24 cidades do estado de São Paulo, 22 delas no interior, uma na capital e outra no litoral, em São Vicente. Essa ampla presença garante ensino de qualidade para mais de 50 mil alunos da graduação à pós-graduação. Estamos entre as universidades que mais produzem ciência no Brasil e temos orgulho de dialogar com as comunidades e compartilhar o resultado do nosso trabalho. Falar da Unesp é falar de todos e todas que ajudaram a tecer essa história em prol de um ensino público, inclusivo e de excelência.
Good morning, everyone. It's a great pleasure to be here. My name is Letícia Lotufo. I am a professor at the Institute of Biomedical Science, USP.
Good morning. It's also a pleasure to be here today. Thank you, everybody, for all these subscribers. And my name is Caroline. I'm a PhD candidate at the University of Paris and in Summit in 1132.
So this morning you have the great pleasure to receive here Dr. Randall Patterson to enlighten us with his knowledge. And before I give him the microphone, I will make a brief introduction of Professor Randall. He is the LS SCAC's Presidential Endowed Professor and the Dean of the College of Pharmacy at the University of Utah. And he's a chemical biologist whose research utilizes high-throughput screening technology to discover new drugs candidates for cardiovascular and nervous system disorders. Prior to his position at the University of Utah, Dr. Patterson was a faculty member at Harvard University during 15 years, where he was the Charles Edson and Elizabeth M. Sanders Chair in Basic Science at the Harvard Medical School, Scientific Director of the MGH Cardiovascular Research System, and also Senior Associate Member of the Broad Institute. So we are all looking forward to hear you, Dr. Patterson. Thank you for accepting the invitation to be here with us.
Well, thank you very much for the invitation. Hello, everybody. Thank you for tuning in. I hope you can hear me okay. It's a real pleasure for me to be able to tell you a little bit about some of the work going on in our lab. And I'm particularly delighted to be participating in this webinar hosted by institutions in Brazil, where I've visited many times and have wonderful friends. Before I begin, I'd just like to disclose some relationships and also acknowledge a wonderful financial support that my lab has had over the years. Most relevant for the talk today is that I'll be telling about some compounds that have been licensed to Kuros Therapeutics, and also want to appreciate the funding support for the National Institutes of Health that supported all three of the projects that I'll be talking about today. I thought I would begin by just reminding everyone that there are two predominant approaches to drug discovery. There's a classical approach that's a phenotype-based approach, where drug discovery begins by selecting a phenotype of interest in cells or animals or tissues, and then compounds are identified that can change that phenotype. And sometimes only much later, years later, is it discovered what the molecular target of the drug was. This is really been the dominant approach to drug discovery historically. But over the last 30 years, this approach has given way to a more modern approach to drug discovery that's a target-based approach, where you begin by selecting a molecular target that you think could be disease-modifying. You create a high-throughput screen using that target in vitro, where you can discover compounds that modify that target. And only at the end of the process do you understand what the phenotype is when you treat animals or humans with that compound. Over the last 30 years, as I said, this has become by far the dominant approach to drug discovery. And in fact, a few years ago, there was a study that found that about 95% of all drug discovery these days is target-based, and only about 5% is remaining from the sort of old classical phenotype-based approach. The big surprise in this study, though, came when they looked at where the new drugs were coming from. And surprisingly, they found that that small sliver of only 5% of phenotype-based screens was yielding about 2/3 of all the first-in-class drugs that had been approved in the decade prior to that. This was really a wake-up call to everyone in the pharmaceutical industry that we shouldn't abandon phenotype-based approaches, that they continue to be a rich source of new first-in-class drugs. So when you think about phenotypes, there's often a difficult decision to be made where you have to choose between trade-offs of phenotypic richness and scalability. So in other words, things like mice and humans have wonderful phenotypic richness and real relevance for medicine. But that comes at very low scalability. You can't test hundreds or thousands or millions of different conditions very easily. On the other end of the spectrum, you have things like cultured cells, which enable you to scale up to many, many conditions being tested at once. But that comes at the cost of reduced phenotypic richness. And things like organoids share some of the best features of both, giving moderate phenotypic richness and moderate scalability. Over the last 20 years, my lab has been very interested in the idea that the zebrafish provide a special combination of scalability and phenotypic richness. Because they're a whole living organism, they offer the ability to look at complex phenotypes, but also to do it at a scale that approaches that can be done with in vitro systems. So when you think about cellular phenotypes, you can think about only looking at a relatively small range of things like cell growth, cell death, cell morphology, gene expression, reporter activities. But when you think about being able to do whole organism screens, it opens the door to a large swath of biology, where you can now examine phenotypes such as insomnia and depression, all the way to cognition and pain. And my lab has been able to apply a chemical biology approach or a high throughput screening approach using zebrafish to a whole range of different organismal phenotypes, including vascular occlusion, iron metabolism, sleep, pain, fertility, and on and on. So for almost 20 years now, the potential of zebrafish screens to discover new therapies and identify drug toxicities has been promoted. And I want to sort of talk a little bit today about, is that potential real? Can we truly discover new therapies that can improve human lives using the zebrafish? I'm actually going to talk about 3 different things today. Here's a brief outline of the talk. I'll first tell you a brief history of the discovery and development of ALK2 inhibitors. This is an older story, but I think it sort of conveys the arc of one of these projects and how you can go from discovery to a treatment that's actually being used in humans. I'll next tell you a much more recent example, just published within the last few months, where we've used zebrafish to discover medical treatment for opioid use disorders. And I'll finally end by something that's not exactly chemical biology or drug discovery, but I think is quite relevant. And I'll describe a new technology that we've developed that is enabling high throughput CRISPR screens to be performed in zebrafish. This first story really begins with a postdoc in my lab many years ago named Charles Hong, who was interested in a fundamental phenotype, how the dorsal ventral axis forms. Charles had done his PhD work at Yale studying Drosophila and how the dorsal ventral axis forms in Drosophila. And so when he joined my lab, he was still interested in this question and wondered if we could discover some new things about the biology affecting the dorsal ventral axis in zebrafish. Of course, a fair amount was already known. For example, it was known that a gradient of the BMP protein, the bone morphogenetic protein, across the early embryo sets up many of the identities of different cell types in the early embryo. And it had been shown previously that genetic disruptions in the BMP pathway could perturb the dorsal ventral axis. So for example, mutations that activated the BMP pathway could cause ventralization, an expansion of these ventral tissues in the embryo at the expense of dorsal tissues. Conversely, if you genetically disrupted the BMP pathway, you would get dorsalization, an expansion of dorsal tissues at the expense of ventral tissues. And so Charles performed a fairly straightforward screen where he took fertilized zebrafish embryos, arrayed them into the wells of 96 well plates, and then he took a library of chemical compounds and placed a single compound in each well. He then simply looked at the fish under the microscope as they developed, looking for compounds that might perturb the dorsal ventral axis. And he screened a relatively small collection of compounds, only about 7,500 compounds, including known bioactive molecules and uncharacterized synthetic molecules. Of those compounds, he found this molecule here, which he called dorsomorphin. And this compound had a profound ability to dorsalize the developing zebrafish embryo. You can see that depending on the time he added it, he could have anywhere from a subtle to a profound dorsalization effect. The ventralmost tissue of the zebrafish embryo is the ventral fin here. And so in the most subtle of cases, he would just lose selectively this ventralmost tissue. And in more severe cases, have more extreme dorsalization. Well, when we looked at this phenotype, we were struck by how similar the dorsomorphin treatment phenotype is to a genetic mutation that had previously been described called loss of fin. That's shown right here. And you can see that this loss of fin mutant causes a fairly specific loss of that ventral fin in the early embryo, just as happens in dorsomorphin treatment. Now, loss of fin is a mutation in a gene called ALK2. And so we hypothesized that perhaps dorsomorphin was inhibiting this target, ALK2. I won't share all the biochemistry that was done to confirm, but we did find that, in fact, dorsomorphin is a potent and selective inhibitor of the ALK2 protein. ALK2 is a type 1 BMP receptor. It, in collaboration with a type 2 receptor, receives the signal from the outside of the cell, from the BMPs, and then transduces that signal through the SMADs to the nucleus. So at about the same time that we discovered dorsomorphin and that its target was the BMP pathway, and specifically ALK2, Chaz came to me very excited with the New York Times in his hand one day. And in it, there was an article with the title, "Finally, with Genetic Discovery, Hope for Escape from a Prison of Bone." This article in the New York Times described a rare and devastating disease called FOP, fibrodysplasia ossificans progressiva. This is a really terrible disease in which these children are born normally, but then over the course of a few years, their soft tissue turns to bone. So here you can see these protrusions of bone in this patient. And after just a few years, these patients get locked in one position, almost like a living statue, as their muscle turns to bone. And in this post-mortem here, you can see that large swaths of muscle have turned to bone. This is a very rare disorder with only about 2,000 patients worldwide. And what this article was reporting on is that scientists at University of Pennsylvania had discovered that this disease is caused by activating mutations in ALK2, the very same gene that is targeted by dorsomorphin. And so we got quite excited about the idea that our compounds, which inhibit ALK2, might be used as a treatment for this rare disease. So many people contributed to this project from there on out, but I want to really highlight three who made enormous contributions, not only to helping us understand the mechanisms of dorsomorphin classes of molecules, but also in thinking carefully about how to develop these as treatments for humans. So I had two physician scientist colleagues at Mass General, where my lab was at the time, Ken Block and Paulo. They made really remarkable contributions to the story that I'm telling you. And Greg Cooney was also our colleague at Harvard Medical School and is a really outstanding chemist. And so we set out to turn our initial hit dorsomorphin into compound that would be sufficiently potent and safe enough to be used as a human treatment. And that began with chemistry. Here's an example of one of the first derivatives of dorsomorphin that we made. You can see that these two molecules share the same pharmacophore, but we've made some changes here to both of these regions of the molecule to produce this new molecule called LDN-193-189. You can see that it's about 50 times more potent than dorsomorphin, and it's more selective too for the ALK2 target. With this molecule in hand, Paulo and Ken Block made a really wonderful mouse model of this disease, FOP, by inserting an activated form of ALK2 and activating it just in one hind limb of the mouse. And what happens, as you can see here in these CT scans, is that after about 15 days after birth, the hind limb begins to ossify. You can see the gastrocnemius muscle here has turned to bone. And by 30 days after birth, you can see that essentially all of the musculature in that one hind limb has turned to bone. But in siblings that were treated with LDN-193-189, you can see a dramatic reduction in the amount of heterotopic ossification. And that may be easiest to see here in these videos. If I can get them to play, hang on. So you can see this is the FOP mouse where one of its legs has completely ossified to the point where it can't use it. And here is a sibling that's been treated with LDN-193-189. And you can see that it retains use of the hind limb. So where to go from there? I thought this was a funny image taken from the NCAT's NIH website showing a simplified map of preclinical development process. As you can see, it's anything but simplified, very challenging process. And so we reached out to and had the wonderful fortune of collaborating with a National Institutes of Health program from the National Center for Advancing Translational Sciences. This program is called TREND, or Therapeutics for Rare and Neglected Diseases. And this is really a partnership between our labs and labs at the National Institutes of Health, who helped us with all of the preclinical development steps needed to prepare these compounds for human testing. Everything from lead compound selection, efficacy testing, toxicity studies, and other IND-enabling studies, and finally, getting compounds that we felt were ready to go into humans. We had many, many people at the NIH who were involved in this project, with really able leadership from Philip Sanderson, who was the TREND project leader. In all, we produced about 1,000 dorsomorphin analogs, and we characterized them, testing for potency, selectivity, pharmacokinetics, efficacy, and safety. The kinds of work that we did together was, for example, to make the compounds more selective. Dorsomorphin itself bound to and inhibited many different kinases. And what we were able to generate were compounds like the one shown here, which is now actually quite selective for hitting just one branch of this kinase family. We also were able to improve various pharmacokinetic properties. So for example, the LDN 193189 compound that I told you about is about 50% orally bioavailable and has a half-life of about 3 hours, whereas we were able to come up with derivatives that were 100% orally bioavailable and had half-lives more in the 10-hour range. And we finally got to compounds that we thought were appropriate for clinical development. Now, this is a challenging clinical trial to perform because there are very few FOP patients around the world, and it's a slow progressive disease with pediatric involvement. And yet, there are some strong advantages, like a very clear connection between the disease mechanism and the drug mechanism. And we were also attuned to the fact that if we could demonstrate utility in FOP, it would pave the way for treating other BMP-associated diseases, like anemia of inflammation. And so at that point, we had the great fortune of licensing these compounds out into a startup company that was led by Jasbir Sihra. This company has a lot of information available on the web, so I won't tell you much more about that, other than to say that these compounds have now moved into clinical trials, and the company had an initial public offering just last year and is now traded on the NASDAQ. So this brings us to the first take-home message that I hope you'll glean from today's webinar, and that is that small molecule screens in zebrafish have successfully identified new therapeutics that are in clinical trials. And this is actually just one example of several cases where zebrafish discoveries have led to compounds with real promise as treatments for human diseases. All right, I'm going to shift gears to more recent work that's going on in my lab right now. And this is work where we've tried to apply this same approach to develop treatments for another major problem that's facing the United States and other countries in the world. And that is an opioid addiction epidemic. You can see here in this graph from the United States that the number of deaths from opioid use disorders and opioid overdoses is going up quite dramatically and is now affecting somewhere in the neighborhood of 70,000 deaths per year in the United States alone. A postdoc in the lab shown here, Gabriel Basset, was interested in trying to see if we could do something about this problem using the zebrafish. And so he developed what I think is a clever self-administration assay in zebrafish. And the assay is as follows. He created these arenas that he could fill with water and have water flowing in one end of the arena and out the other, so that the water was always refreshed. In the arena, he then placed 2 platforms, each monitored by a camera. And if fish were placed in the arena and they swam over the inactive platform, it's recorded as an event, but nothing happens. If the fish swim over this other platform, the active platform, however, a light flashes, it's recorded as an event, and a small puff of liquid is delivered to the platform through a tube. All of this is being monitored and automated using motion tracking software and off-the-shelf robotic systems and a Raspberry Pi computer running a custom Python script. So using this platform, he then trained fish, zebrafish, adults, for about 50 minutes per day for 5 days. And he used an opioid called hydrocodone or Vicodin. And this is what he found. If he used water for the conditioning and not an opioid, he found that on the first day of trials, the fish explored quite a bit, and they swam over the active and inactive platforms the same number of times, about 800 times. And over the subsequent days, the fish became less curious and swam over both platforms less, but still the same number of swims over the active and inactive platforms. But when he added hydrocodone to the solution that was delivered to that platform, what he saw is as follows. The fish, within just a few days, rapidly lose interest in the inactive platform, hardly ever swimming over it. Instead, they spend all of their time swimming over the active platform to deliver more and more doses of opioids. Here's what that looks like. You can see that these fish, which have been conditioned for 5 days, almost never go over to the inactive platform, and they spend all of their time swimming around the active platform and never going more than a few seconds without swimming over the platform to get another dose of opioids. I won't show you the detail, but we found that these fish who had been conditioned to self-administer opioids will engage in risky behaviors to seek a dose. They'll show signs of withdrawal when they're denied opioids, and they can be recovered from the effects of opioids by treatment with naloxone, which is used also to treat humans who have overdosed with opioids. With this assay in place, we were able to do a relatively small scale screen of just over 100 drugs that we thought might be interesting to test in this model. And of the 100 compounds tested, we discovered one that had a profound ability to reduce opioid seeking in the zebrafish. The compound that we discovered is shown here. It's called finasteride. This is a compound that is known to be a 5-alpha reductase inhibitor, and it's used clinically in humans for 2 things. First, to treat baldness, and also to treat males who have an enlarged prostate. What was interesting about this is while finasteride could reduce the interest in zebrafish for seeking opioids, it didn't seem to interfere with their interest in seeking other things like food. Here we've used that same arena to test whether it would have an impact on hydrocodone seeking, and you can see that it dramatically reduces hydrocodone seeking. But if we used food as the reward instead of opioids, it had no impact on their seeking of food in this system. We've also tested this now in rats in 2 models, one using an injected version of morphine, self-administration in rats, and another an oral fentanyl model. And we found that in both of these models, treatment with finasteride could dramatically reduce the amount of opioid consumption in these rat models. And so this got us really interested in thinking about whether we could repurpose this existing clinical drug for treating opioid use disorders. We also have been trying to understand mechanistically how this works. I mentioned that finasteride is an inhibitor of the 5-alpha reductase enzyme. 5-alpha reductase is involved in the metabolism of testosterone to dihydrotestosterone, but it also converts other neurosteroids in the brain from one compound to another. And we've been interested in trying to figure out which neurosteroid might be mediating this change in opioid seeking. And so we've performed steroidomics in zebrafish and also in mice. And we've found that the most significantly changed neurosteroid is this one shown here, DHEA. We've found that they're significantly altered in the brains of fish and mice following finasteride treatment. And that treatment of zebrafish with DHEA alone is sufficient to reduce opioid seeking in our model. And so we think that it is changes in the level of DHEA in the brains of animals that's reducing their desire to seek opioids. So that leads to the second take-home message, which is that finasteride reduces opioid seeking through changes in neurosteroids, specifically DHEA. And our hope that this compound or a related one could be used to treat opioid use disorders in humans. And we're now turning our attention to trying to develop a clinical approach for testing this hypothesis in humans. I'll now move on to the final little vignette for today's webinar. It's again not directly drug screening, but instead involves CRISPR screening, and I think is hopefully of interest to this audience. And also, I hope you'll be able to see that it can provide some real benefit in terms of identifying the targets of new small molecules that are discovered in zebrafish screens. All of this work was really led by a wonderful postdoc in the lab, Saba Parvez, shown here. Now, I'm sure this audience, I'm sure all of you are very familiar with the impact of zebrafish in the field of forward genetics. Forward genetics has been around for decades in zebrafish, and has been a really powerful approach for illuminating gene function. That has wonderful advantages that it can be done in high throughput. Theoretically, you have the ability with forward genetics to examine the entire genome of the zebrafish and discover new genes involved in a whole range of different processes. One of the real downsides, however, is that these mutations, whether they're chemically induced or insertional mutations, are created randomly, and there can be quite a bit of work involved in figuring out which gene has been mutated. More recently, just about a decade ago, reverse genetic approaches became accessible in the zebrafish, including zinc finger nucleases, talons, and CRISPR-Cas. And my lab has had the great fortune, along with our collaborators Keith Jung's lab and Joanna Ye's lab, to be involved in the development of all of these technologies in the zebrafish. This has some great advantages, being that you can now make targeted mutations in virtually any part of the genome that you would like, but it has one major downside, and that is that it's low throughput. These approaches really only enable you to target a single gene at a time. And although there have been some reports describing dozens or even more than 100 genetic mutations, each of these was made simply by brute force scaling up of the methods that are used for making individual mutations. And there hasn't been a way of performing high throughput, scalable CRISPR screens in the zebrafish, or really in any animal for that matter. And so we wanted to try and address that and have developed a technology that we call mic drop. It stands for multiplexed intermixed CRISPR droplets. And we think that it's going to be a really useful approach for performing high throughput CRISPR screens. Let me tell you a little bit about the technology. It begins by designing and producing guide RNAs for any number of genes in the zebrafish genome, anywhere from a few to theoretically thousands of different genes or the whole genome. A library could be produced. And so here each well represents a different gene that's been targeted. And typically we multiplex this or target about 4 different guide RNAs to any gene of interest, just to make sure that we've truly disrupted the function of that gene. We mix with the guide RNAs the Cas9 protein and a DNA barcode that matches the identity of the gene that's being targeted. We then use a microfluidics approach to generate nanoliter-sized droplets where you have an aqueous droplet with the guide RNAs, the barcode, and the Cas9 protein all encapsulated within a fluorocarbon oil. These droplets can then be intermixed, combined into a library of droplets with as many different genes as you want to intermix. And you can then load those droplets into a needle and use that needle to inject a single droplet into each zebrafish fertilized embryo, 1 droplet per embryo. And you can then raise the fish in a group to whatever age you are interested in, and then screen them for the phenotypes that you're interested in. When you find a phenotype of interest, you simply collect the barcode out of those fish, and you'll know the identity of the gene that was targeted. So what does this look like in practice? Well, first I'll tell you a little bit about the production of the droplets. What do they look like? Here's an example of these droplets. Each one is about a nanoliter in volume. They're quite uniform, and you can produce thousands of them in just a few seconds using a microfluidics device. And then after you have mixed them together, you get something like this. This is a typical glass needle, such as most of you probably use for injecting zebrafish. And you can see it's now been filled with thousands of droplets. Each of these have been pseudo-colored just to help you sort of distinguish them. But you can see that you can now have many, many droplets in the needle, with each droplet targeting a different gene. Once that needle has been filled, hopefully you can see this video, you just inject almost like you would normally. You inject a single droplet into one fertilized egg. You then expel the oil that is in between the droplets just into the water. And then you inject the next droplet into the next embryo. And you expel the oil until the next droplet is up to the tip of the needle, and you can move on like that. So with this method, you can now, with the single needle, in a single morning, you can inject hundreds or more different guide RNAs into different zebrafish and just raise those fish en masse. We find that there's very little mixing of droplets. So here's an experiment where we took guides targeting 3 different genes, and we mixed them together in equal proportions and injected individual droplets into zebrafish embryos And then we looked at them, and we found that about a third of the injected fish were albino-only phenotypes, a third were silent heart phenotypes, and a third were cordino or ventralized phenotypes, with very few fish that showed a mixture of more than one phenotype. We found that these droplets are really quite efficient. Somewhere in the range of 90% of the injected animals exhibit the phenotype as shown here using the tyrosinase gene, and that the efficiency is essentially equivalent to what you would get if you injected just the guide RNAs and Cas9 protein by itself. And these droplets are actually surprisingly durable. Just by refrigerating them, you can hold on to them for up to a month with only modest reduction in the efficiency of phenotype production. In terms of barcode recovery, we used DNA barcodes. There are, of course, other approaches that could be taken. And we found that injection of these droplets even with the oil and the barcodes and the guide RNAs and the protein, that we had very good viability, somewhere around 95%, even with relatively large amounts of barcode included. And we found some percentage of deformed embryos. And we found that we could recover the barcodes for at least 7 days after the initial injection for very robust identification of the genes that we had targeted. So then we did some other tests. For example, we took a bunch of scrambled guide RNAs and we doped in a 50-to-1 ratio, scrambled to tyrosinase or cloche guide RNAs. We then injected them into zebrafish and scored the phenotypes. And what we found was the expected frequency for both of these was about 2%, which is what we expected for a 50-to-1 ratio. So we then wanted to try and see if we could use this approach to discover some new genes. We used a bioinformatics approach working with Chelsea Herdman in Joe Yost's lab. And we looked for genes that were enriched in the heart and also enriched in embryos compared to adults. And we found about 465 genes that passed that filter. And we selected from those 188 genes that had very poorly annotated functions. And we made a library with these 188 different genes and performed a mic drop screen. And what we found was more than a dozen new genes that had cardiovascular phenotypes in these animals. We also performed a number of positive controls using genes that are known to be important for cardiovascular development. You can see that the range of penetrance here was anywhere from, single digit in a few cases to 100% in other cases. I'll just sort of briefly run through some of these phenotypes. We discovered that CLEC19A causes a developmental defect in the heart characterized by a lack of looping and an expansion of this AV canal, and a valve defect, which you can hopefully see here on the left is a wild type heart beating, and CLEC9 mic drop morphant or crispant here with a regurgitation of the blood back and forth in the atrium and ventricle due to defects in the valve formation. We found a couple of genes that are involved in cardiac rhythm, including this one, GSTM.3, in collaboration with Manu Birins and Callum McRae's lab. We were able to characterize some of these arrhythmia mutants and show action potential duration deficits in these crispants. And here's another example of the ALAD gene, which causes a profound porphyria. Many of these phenotypes we were able to rescue by simply injecting the targeted gene as an mRNA, confirming that in fact these phenotypes are caused by mic drop disruption of function of the intended target. So I hope I've conveyed that this mic drop platform is a robust platform for in vivo functional genetic screens, both morphological and behavioral phenotypes or any other phenotype that you might want to study. It can be scaled up to target thousands of genes at the same time. It's not limited to targeting coding genes. You can, of course, code or target non-coding elements. You could use the same kind of approach for CRISPR-A and CRISPR-I, or knocking down RNA with Cas13, and probably many other applications. Another application that's of great interest to us is using this technology as a target identification platform. As you may know, once you discover new compounds using a phenotype-based approach, one of the real challenges is discovering the target of that, and we think this might be a useful approach there. Here's an example of a compound that we discovered a few years ago called Optivin. This is a compound that binds to an ion channel called TRPA1, and when you shine light on it, it opens the channel and activates the nerve. And so without Optivin, fish don't really respond to purple light, but in the presence of Optivin, you can see this profound response to purple light. And if you've knocked out TRPA1B, then you don't get that response. And so we performed a proof of concept experiment where we doped in a small number of mic drop droplets that target TRPA1B into a large number of scrambled droplets in a 20-to-1 ratio, injected them into zebrafish, and then arrayed them into 96-well plates where we treated with Optivin and flashed purple light on them. And we found that out of this plate of zebrafish, there were 3 that did not respond to the combination of Optivin and purple light. And so we pulled those fish out of the wells, recovered the barcodes, and all of the barcodes were indicated that those fish had been targeted for TRPA1B knockout. So the final take-home message here is that mic drop technology is a new technology that enables large-scale CRISPR screens in zebrafish with applications ranging from gene discovery to target identification. With that, I'd like to thank once again the key contributors to these projects. I think I've mentioned most of them along the way, but Charles Hong was really the initial discoverer of dorsomorphin and leader of the dorsomorphin project, with really important contributions from Paulo, Ken Block, Greg Cooney, and also Chetna Satchidanandan and Philip Sanderson and the TREND team, with wonderful support from the NIH. And the opioid project, Gab Basse was the leader, with Marco, Roberto, Ryan, and Paul contributing to the rodent models of opioid addiction, and Nilesh performing the steroidomics. We've had funding from the National Institutes of Health of that project too. And for the mic drop project, Saba really has led the project with informatics support from Chelsea and Joe, characterization of the arrhythmia mutants by Manu and Callum, and intellectual contributions from Joanna Ye. So with that, I'll thank you for your attention.
Thank you very much, Dr. Peterson, for this beautiful presentation. I believe that the discussion of how we should choose between the target-oriented approach and the phenotype-oriented approach in drug discovery is like it's not the point. The point is that we should be using both, and you just gave us a beautiful example how the zebrafish can come and put these two things together and give some amazing results. But I think you have quite a few questions from the audience. So I will, Caroline will start with the first one. Please, Caroline.
Thank you very much, Professor. We have a lot of questions. I'll try to, I don't know if we have time for all, but we start with the first one from Mônica Rodrigues Ferreira Machado from the University of Federal University from Rio de Janeiro. She said, "Nice presentation, Rondo. Have you seek for effects of finasteride to enhance estradiol in brain?"
Thank you very much, Monica. Great question. The steroidomics that we did not reveal a very significant change in estradiol levels in the brain. Yeah, I was a little bit surprised by that, but that's not one of the changes that we see.
Okay, thank you. And then we have the questions from Tainara Maria Moraes from Unicristo, and she said that I find the work on opioids very interesting. With so many results, do you think in the future, is it possible for the community to focus on using this model as the main one?
Thank you very much, Tainara, for that question. I'm not sure if I can ask a clarifying question, but do you mean using zebrafish as primary compared to the rodent models? If that's what you're asking, I think that would be wonderful. I would love to see that. I suspect that most of the scientific community still has more faith in the rat model. That model has been used for, of course, many, many years. And even in publishing our work, we felt it was important that we confirm our compounds had activity in these established rodent models. And I think that's a model that we've used for many of our projects. We do the high throughput work and the discovery work in zebrafish, and we often move then to a mammalian model for confirmation. And I think one of the great things that comes from that approach is that if you can show conservation of effect from a non-mammalian zebrafish all the way to a mouse or a rat, that suggests that this phenomenon is very robust and could be probably extended further to humans. If you've already gone from fish to a rat, the leap now to go to humans is relatively small. So I think there's real power in showing activity across multiple species, and it gives confidence to people that this has real relevance for humans as well.
Thank you. We have another question, and now it's from Shadow Mix. What is a risk behavior for a fish seeking opioids? Have you seen to what extent novel experiences may compete with addiction behaviors? Like, for example, meet a new fish/environment as an out-drug dose.
I love this. Great question. Very, very interesting. So the risky behaviors are, one of the riskiest things for zebrafish is to swim into shallow water. And for those of you who have, paid attention, you'll notice that your fish, particularly when they're stressed, do not want to go into shallow water. And so if we put a high platform where the fish have to swim up into shallow water, they won't do that normally. But if we've conditioned them to develop, to self-administer opioids, now they'll swim up into shallow water over a platform to get a dose. So that's what we define as risky behavior. There was a second part of that question, but I'm afraid I've forgotten it.
Yeah, it was meet a new fish/environment as an out-drug dose.
Yeah, humans who become heavily addicted to some substances will seek the drug rather than other rewarding activities in their life. And so we have thought it would be quite interesting to create a choice assay where then the fish have to choose between opioids and a social interaction, or opioids and sex, or opioids and food, and see if they similarly would choose the opioid over other rewarding activities in their life. But we haven't done that. I think that would be a really interesting thing to do.
We do have another question from Veronica Lombardo. She says that, excellent talk. I have a question. Using the self-administration test, it's possible to study the reaction of the zebrafish to toxic molecules. Did you use that approach?
Well, that's a really interesting idea, Veronica. We haven't tried that. I think the closest thing that we did was to add into the opioid mix a substance that we thought would be aversive to them, to see if they would be willing to swim to the molecule and take the opioid, even if it was, a molecule that normally would repel them. But I think you're right that the same kind of strategy could be used to look at other behaviors, including responses to toxic molecules. Very interesting idea.
Thank you.
There's one more question from João Mamed. Amazing presentation, Professor Randall. With finasteride and DHEA, you focus only in seeking behavior, or there is also something that influences the effect caused by the opioids?
Great question, João. Thank you for that. We certainly haven't looked at everything, but we've looked at a couple of things about the opioid effect, and in particular, whether the finasteride would interfere with the analgesic effects of the opioids. And so we've used a number of pain models in both fish and in rats. Most prominently, perhaps, is a neuralgia model where we create pain in the rats and test the ability of opioids to alleviate that pain. And we find that the finasteride has no impact on the analgesic effects of the opioids. That's really important because I think ultimately it would be one cool application would be to mix finasteride with opioids in such a way that it reduced the addiction liability, but didn't interfere with the ability of the opioids to do what they're supposed to do, which is alleviate pain. Great question.
Thank you, Professor Peterson. I'm afraid that we should stop now. We still have a lot of questions. Everybody loved the talk. So thank you again for joining us this morning. But now we have to go for the other talk. Thank you. I don't know if Carolina wants to say some words.
Just thank you for your presence and your sharing your knowledge with us. It was really interesting. And thank you very much. Thank you for for the great questions as well.
And now we will
thank you. It was a real pleasure.
Sorry. So now the organization is telling us that we will be back in a few minutes, okay? See you.
Trabalhamos diariamente para criar soluções e equipamentos para a comunidade científica, contribuindo com o avanço da pesquisa biomédica latino-americana. Nossa missão é maior do que somente fornecer equipamentos. É proporcionar segurança com atendimento de qualidade e, principalmente, com muito respeito e atenção. Por isso, investimos em alta tecnologia e buscamos manter relacionamentos duradouros. Nosso compromisso é ser um parceiro confiável que compreenda as necessidades, a realidade e as condições de cada cliente para assim oferecer a melhor solução sempre. Entendemos os benefícios da pesquisa científica para a humanidade e isso nos estimula. Se hoje temos melhor qualidade de vida, maior longevidade, se vencemos um câncer ou fazemos uso de um remédio para dor de cabeça, é porque o avanço da pesquisa biomédica nos permite. Confiamos no trabalho dos pesquisadores, na ciência e na comunidade científica. E nos orgulhamos em fazer a nossa parte. Assim como você, somos apaixonados. Ciência é o que nos move, porque para a Alesco, pesquisa é para a vida.
Well, now for the next talk, I'm honored to invite Professor Stephen Renshaw. Professor Renshaw is the Sir Arthur Hall Professor of Medicine at the University of Sheffield in England, combining clinical work in respiratory medicine with research into the molecular controls of inflammation resolution. Professor Renshaw has a special interest in rare and complex interstitial lung diseases and delivers acute respiratory care at the Sheffield Teaching Hospital NHS Trust. His key research achievement is the development of innovative transgenic zebrafish models to add understanding of innate immunity. Thank you very much, Professor, for being here with us today.
Hello, and thank you so much for the very kind introduction. So I think you can see my slides there, so I'll make a start. Thank you very much. That was a fantastic talk we've just heard from Professor Peterson, and my talk is going to be a little different. I'm going to talk about some work we've been doing on inflammation and really give you a view from the other side of the Atlantic. So I'm going to talk about 4 stories today. I'm going to talk a little bit about how we use the zebrafish to help us understand inflammation, and show 3 specific research projects, one around neutrophil swarming, one very briefly showing some mechanisms of inflammation resolution, and then finally I'm going to show some clinically, what I hope is clinically relevant studies on cystic fibrosis. So as you heard in that very kind introduction, I'm a respiratory physician. I still see patients with respiratory diseases here in England. And to give you a bit of an idea of the sort of things that we see, this graph shows the admissions to respiratory medicine in 2004, although things have been a little different in the last year or so. They're still largely the same as these days. And one of the key things about this is that pretty much half of our admissions come from infection. And obviously we had a little spike earlier in the year when it was a little bit more than that. But the other half of these conditions are essentially inflammatory conditions. And I think this tells us that the innate immune system, which we rely on for protection against infection, is really very delicately balanced and can really cause problems if overactivated or indeed underactivated. And just to show you the sorts of conditions that I deal with, I'm just going to show you a few chest x-rays. So I'm aware most people won't be familiar with looking at chest x-rays. What I want to show is that this is relatively normal lung here, the heart in the middle, the right lung on the other side. What you can see are these patchy shadows here in both lungs, and this is an example of COVID pneumonitis, where there is neutrophilic inflammation in the lungs. And there's some really interesting work coming out now with the role of neutrophils in the pathology of COVID pneumonitis. This is the acute respiratory distress syndrome, and you can see, I hope here, that the normal black appearance of the lungs is completely replaced by this greyish mottled appearance, and this is fluid and neutrophils filling the alveoli in this patient with acute respiratory distress syndrome. In contrast, this is a chest x-ray of somebody with chronic obstructive pulmonary disease, and what I hope you can see is these lungs are much bigger and much blacker than the previous ones, just to show by comparison again. And this is because the normal lung tissue has been destroyed by the action of neutrophil proteases, leading to degradation of elastin, collapse of the normal architecture of the lung, and air trapping, allowing the lungs to hyperinflate. And this is what that looks like at on an electron microscopy level. This is normal alveolar structure in the lung, and you can hopefully see this very thin gas exchange membrane between these air sacs. And what happens in COPD is that these little air sacs are destroyed by the action largely of neutrophil proteases, and you get this accumulation of mucus and inflammatory cells here in the lung. And finally, this is not a chest x-ray anymore. This is a reconstructed CT scan. This is a slice through people. And what you can see, hopefully, here are these big dilated air spaces here that you shouldn't get, big cystic lesions within the lung, full with fluid and infection. And this is a patient with cystic fibrosis. So in all of these conditions, the action of neutrophils is really important. And this is the cell that I have become interested in. So these are neutrophils. These are actually zebrafish neutrophils, but they look very similar to mammalian neutrophils. And they're doing what neutrophils are there for, phagocytosing and killing bacteria. So you can see these bacteria changing from green when they're alive to blue, which is when they've been killed by reactive oxygen species. So this is what neutrophils are for. They protect us against infection, and they do a very, very efficient job of that. But what you can also see in this movie here, this is a membrane-tagged blue fluorescent protein within neutrophils. You can see that neutrophils contain lots and lots of these granules within them, and neutrophils are called granulocytes for that reason. And each of these granules contains reactive oxygen species and antibacterial peptides and proteases. And these proteases and reactive oxygen species can be delivered into tissues during inflammation and can contribute to tissue damage. And it's these proteases that are responsible for the tissue damage that we've seen in the sort of lung conditions that I've described. So neutrophils are an unusual cell. They are produced at rather high amounts by the body. So in mammals, they're produced in the bone marrow, and we produce about 1 million neutrophils a minute. And that can go up dramatically during times of infection or stress. And most neutrophils will circulate for a relatively short half-life. There is some debate about this, but it's probably in the order of a day or so. And most of them then will return back to the bone marrow to be recycled. But if they encounter an area of tissue damage, shown here in this cartoon, there's activation of the endothelium, and neutrophils undergo the sequential process of rolling, firm adhesion, and then transmigration into the tissues. So in this cartoon here, you can see the tissue edema, tissue fluid, and the neutrophil in the tissues. And it's in the tissues that neutrophils can cause the damage, where they can degranulate and perpetuate the inflammatory response. So in order for inflammation to resolve, neutrophils must be removed from the tissues. And one of the ways this can happen is by the apoptosis of neutrophils, which turns off the pro-inflammatory function of the neutrophil and also marks it for recognition and uptake by macrophages. And the act of taking up an apoptotic cell changes macrophage phenotype to a phenotype which is more suitable for tissue healing and homeostasis. So by this process, tissue homeostasis is restored. So this is the process that I was interested in early in my research career, and it was these sorts of questions that I moved into Zebrafish to help understand. So I wanted to understand initially how these signals that neutrophils encounter at inflammatory sites, these cytokines, hypoxia, and bacterial products, could influence inflammation by turning off apoptosis and preventing this normal resolution process. And when I was setting up my lab, there were really only two ways to do this. One was to look in human neutrophils, which you can see in the top left, and the other was to look in mice. And human neutrophils are great. They really are the cell that we're interested in, and you can get them out of peripheral blood from volunteers very easily, but you can do nothing with them. They survive in vitro for less than 24 hours. They can't be genetically manipulated even today. And they're very, very sensitive to changes in the environment, and things that you do to them, even just in terms of pipetting them too vigorously, can cause activation and disrupts their normal signaling pathways. Mice, on the other hand, are very useful. They can be genetically manipulated, but it is still very difficult to see inflammation, particularly during the resolution stages, even now, although there have been significant advances in intravital imaging over the last 15 or so years. So I wondered when I set my lab up whether we might be able to find a model in between, and it will come as no surprise to you that we use Zebrafish as our principal model. The main reason for this is that Zebrafish development is so rapid, and the rapid external development of Zebrafish that you can see going on before your eyes here is one of the real benefits of the Zebrafish model. And this meant that developmental biologists got excited about the model and worked out the imaging and the genetics, and that meant the model was available to people like me who were interested in using it for inflammation. So really, over a matter of hours, the zebrafish moves from being a single fertilized egg into being a structured embryo. This is 12 hours coming up here, and you can see already the spine and the segments forming here, and the eye. And really, by the time we get to sort of 18 hours, we're starting to get the first immune cells formed. And by 24 hours, both neutrophils and macrophages are formed and fully functioning, protecting these organisms against infection. So around the time that we start to do our experiments, really we're dealing with a fully functioning innate immune system. The adaptive immune system forms later, but the presence of the innate immune system means that we can study this really from very early stages. And zebrafish share their immune systems with the rest of vertebrates in many ways. So if we go back over evolutionary time, perhaps some 450 million years ago, to the time that cartilaginous fish branched off from the tetrapod lineage, around this time we had the full development of innate and adaptive cellular mechanisms, and many of the humoral mechanisms as well, such that sharks are the first animals to have separate T and B cells, and granulocytes and lymphocytes had appeared sometime before that. So well before the origin of the bony fish, which would include zebrafish, the cellular components, at least of immunity, were very strongly in place. And although there have been some molecular changes due to the different pathogens that different mammals face, the basic principles of immunity are well conserved. And specifically looking at neutrophils have many of the morphological features of human neutrophils. They share many of the biochemical features, and they have a number of the functional similarities as well. They are phagocytic, they make neutrophil extracellular traps, and they have this granular poietic response to infection, with the numbers increasing dramatically. So one of the very earliest things that I did in the lab when we wanted to study neutrophils was to make this transgenic line, which labels neutrophils with green fluorescent protein. And following sterile tissue injury, you see an accumulation of neutrophils at the site of the wound, and then you see inflammation resolution with many of these neutrophils leaving. But the great thing about the zebrafish is that they are really amenable to the imaging. So this is a light sheet movie showing the distribution of neutrophils and macrophages in transgenic embryos. And we can zoom in and see really unprecedented detail of the behavior of these cells during the inflammatory response. And one of the things that we've seen, this isn't a particularly good example, I have a better example, although I do apologise for the green-red for those of you who aren't able to see that clearly. But this is an example of a neutrophil undergoing apoptosis in vivo and very rapidly being recognised as such and taken up by a macrophage. So neutrophil apoptosis does happen in vivo, although, as we'll see in a moment, that isn't the whole part of the story. And we're also able to look in more detail at intracellular signaling pathways now, rather than just labeling the whole cell. So these are neutrophils where we've labeled the actin in red, and this is PI3 kinase signaling, the pH domain of AKT. I'll just play that again. And what you can see are these are bacteria that have been taken up, and we can see the localization of PI3 kinase signaling around phagosomes here within the cell, and this actin rich structure as the neutrophils move. So the first story I want to tell using this system is one about neutrophil swarming, which is a relatively recently described phenomenon that we've become interested in recently. And the work I'm going to show has been performed by a really talented PhD student and then postdoctoral scientist, Hannah Isles. So this movie is one that we generated many years ago, which just shows one of the advantages of the zebrafish, that you can see every single neutrophil during the whole of the inflammatory response. And I've been showing this at talks for many, many years now. But one of the things that Hannah said to me is, she said, well, look, there's a swarm there. And it turned out that she was right. So in terms of what I mean by a swarm, This was described first by Tim Lammerman in this lovely Nature paper, and he performed the experiments in mice, and he described this phenomenon whereby neutrophils attract other neutrophils by relay signaling that leads to dense accumulations of neutrophils in tissues. And he showed that by looking at distance time plots, that the early neutrophils might move rapidly towards the wound over time, but later on there's this very distinct second phase of neutrophil recruitment, which is very, very coordinated, very direct and rapid recruitment of neutrophils to the wound. And they did these experiments in a really elegant setup where they took bone marrow from a range of mice, including a range of different knockout mice. They purified the neutrophils and injected them into the ear of another mouse. And then they set the mice up for intravital imaging and did a little laser wound on the ear. And that caused this accumulation of neutrophils, initially just small numbers. They showed that there was a stage where they thought that they saw pioneer neutrophil death, the pioneer being the first neutrophil to arrive. And then there's a second larger amplification wave where they get relay signaling through leukotriene B4. And the experiments that showed this really very elegant. They took a whole series of knockout mice and showed that when they transferred in the neutrophils missing the enzymes necessary to make leukotriene B4, that they no longer saw this big second wave of killing. And they subsequently then see this phase of tissue aggregation and tissue remodeling, and finally swarm resolution. And although my interests are in inflammation resolution, what I'd really like to get to is this stage of looking at swarm resolution. Really, we felt that the zebrafish had something to offer in looking at these early stages, and we wanted to try and study these and see whether or not we saw swarming in the way that we thought we did in that movie. So just for comparison, this is the mouse data from Tim Lammerman showing the neutrophil recruitments to the wound, and we showed a subtly different phenomenon, but similar. So we have a linear wound here. We don't have a single point wound, and the neutrophils could, in theory, go anywhere. But it turns out that they don't. A single neutrophil arrives, and then other neutrophils are recruited to it in a very similar way. But because this isn't a point wound, it's a linear wound, and because these are not exogenously added neutrophils, these are endogenous, we felt this was a particularly useful model for understanding this process. And in terms of characterizing the process, we showed that the neutrophils tended to accumulate over a period of about 4 hours after injury, and occasionally we saw swarms that where neutrophils would accumulate and then the swarm would dissipate over time. But most zebrafish that we studied had a persistent swarm following injury to the tail fin. And we saw a similar phenomenon to this late phase recruitment here. The timescales are quite different here. This is 30 minutes, and 30 minutes is really the beginning of this one here. But you can see that these more distant neutrophils are attracted again with this very rapid accumulation as time goes on. And we wanted to also investigate this leukotriene B4 dependence of the phenomenon. So what they showed is that this later phase was really almost completely abolished by the loss of the leukotriene receptor here. And what we showed in zebrafish was that they expressed the leukotriene A4 hydrolase, the enzyme that makes leukotriene, and also the receptor, particularly BLT1. And if we knock down either of these using transient CRISPRs, we could show that the initial recruitment was normal, but the second phase of recruitment was missing, and neutrophil numbers declined much earlier in the zebrafish, suggesting this is dependent on leukotriene B4. But the thing that is really good about this model is that we can take a swarm here and we can wind the clock backwards and see what happens at the beginning of the swarm. And we found that each swarm was started by a pioneer cell. And that doesn't just mean it's just the first cell there. This cell is different in its behavior to the other cells. These cells arrive and they sit there doing nothing for between about 5 minutes and about 80 minutes, with an average of just under 40 minutes. So these cells are different to other cells. They pick a spot, they sit there, and they seem to do very little. As you can see from the image here, they're rounder, and we can confirm that significant numbers of pioneers and non-pioneers, and they don't move, so they just they sit there not moving very much, and this happens at the beginning of each swarm. So we were able to confirm these phases where a neutrophil will move into the site of a wound, this scouting phase. It will sit there and do nothing for a significant time while it's initiating the swarm, and then eventually there'll be aggregation of neutrophils in a leukotriene B4 dependent fashion to that site. So we wondered whether this initial neutrophil is apoptotic. It's not moving. It's rounded. So we had a transgenic line that we'd made that's a fret-based reporter, a CFP-YFP fret, where these two molecules are joined by a caspase sensitive linker sequence. So during caspase activation, during apoptosis, this is cleaved and the fret signal disappears. And what we could see is that these neutrophils around a swarm here maintained this fret signal, this green. So a pioneer neutrophil here never lost the fret signal. In this case, it's shown as purple. And we can show that when a neutrophil does undergo apoptosis, so the fret signal here is lost, that there is no swarm forming. So we really don't think that that process is apoptosis. And while we're trying to work out what that process was, Hannah observed this strange phenomenon where neutrophils would seem to eject material from within them into these sort of balloon-like structures that emerge from the neutrophil. And that's what this looks like on the stills. So we wanted to investigate what those were in a little bit more detail. So Hannah made a line to label neutrophil extracellular traps, nets. So these are a chromatin structure that neutrophils extrude upon stimulation, and it's thought to be an antibacterial structure. The chromatin is in itself antibacterial, but it's also studded with the antibacterial proteins and peptides that the neutrophils make in their granules, and it has been shown to have roles in host defense. So this line labels chromatin. It's a histone 2A line, and what you can see is as the neutrophils extrude this net here, it is positive for well, the extruded structure is positive for chromatin, suggesting it may well represent a neutrophil extracellular trap. And in order to see whether these are functionally important, we inhibited a number of elements that are necessary for generation of neutrophil extracellular traps. So probably the most important are neutrophil elastase and gasdermin D. And when we inhibited either of these with chemical inhibitors, we saw a reduction in swarming. We didn't see any reduction with caspase inhibition, which would fit with this not being a caspase-dependent apoptotic process. And we also saw a reduction when we knocked out myeloperoxidase, and this is using transient CRISPR again. So it looks like that all the things that are necessary for net formation, when you target them, you get a reduction in swarm formation, suggesting that these swarms may be functionally important in producing swarms. And we wanted to test whether it was the pioneer neutrophil that became the center of a swarm. And to do that, we used this photoconvertible protein Kaidi, which you can change from a green to a red fluorescence using the laser on a confocal. And we showed using that if you pick a cell that you think might become a pioneer cell and photoconvert it, and you follow it over time, you can see that cell releasing these extracellular structures in the same way during the initiation of net formation. So we think that we're seeing we're seeing swarms in zebrafish, and that these occur by the recruitment of pioneer neutrophils to the wound, which then releases local chemoattractants, potentially related to the process of net formation, and that these then cause the aggregation of swarming neutrophils amplified by leukotriene B4. And we're really interested now to use this model to try and understand the resolution of nets and how those neutrophils will eventually move away from the sites of inflammation. So I just wanted to tell you a little bit of what we've been doing to look at inflammation resolution. And this is really not very detailed, but because this work has all been published. And I should say, actually, we've just had the the swarming work accepted for publication today in eLife. So this is the movie again that I showed earlier, showing the every neutrophil in the fish during inflammation. And what was very clear in this is that we don't see neutrophils moving into inflammatory sites, undergoing apoptosis, removed by macrophages, and that being the only process. A lot of these neutrophils seem to traffic backwards and forwards to the wound. And we did wonder whether or not there were other features at play. And in fact, Anna Huttenlocher, when she published similar work, had described that neutrophils do leave the wound sites by reverse migration. And we set up an assay using this CAIDI system again, where we let the neutrophils recruit to the wound, we photoconvert them from green to red, and then we follow where those wound neutrophils go. And you can see a number of these neutrophils leave the wound by reverse migration. And when we started to get really interested in this was when we turned on hypoxia signaling, which is a profound pro-inflammatory stimulus for neutrophils. We found that we turned off reverse migration. So in this movie, we've activated the HIF pathway chemically. And what we can see is over the same time course as the picture to the left, there are virtually no neutrophils which leave the inflammatory site. And we also showed that this wasn't because they weren't moving, they were moving just as much as the other ones, but they just chose to move up and down within the wound rather than moving away. And this is just the quantification to show that with DMSO, which is our control treatment, neutrophils will leave the wound over time. And with DMOG, which is the chemical activator of the HIF signaling pathway, we suppress that process. So this was something we thought was very interesting. And at the same time, we were following Randall Peterson's lead and doing some chemical genetics work in zebrafish. And we did an assay based around our inflammation work, where we injured the tail fin of transgenic zebrafish. We waited for neutrophils to accumulate at the wound, and then we arrayed them in these 96-watt plates with chemical libraries. And our negative control looks like this. We still have neutrophils accumulated at the wound site. And the positive looks like this, or HIT compound looks like this, where we have no longer any neutrophils at the wound. So we have driven inflammation resolution. So one of the best hits that we had was called tangenone 2A, which is a pure molecular entity. It's a compound derived from medicinal sage used in Chinese medicine. And we were able to show that this has an effect on human neutrophils, a dose-dependent increase in apoptosis. But importantly, it also overcomes the survival effect that you see with pro-inflammatory signals such as GM-CSF. This normally suppresses levels of apoptosis, and in the presence of tangenone, it doesn't suppress apoptosis. In fact, there's a small increase in apoptosis, which is maintained whether or not you have GM-CSF present. And this is what human neutrophils look like with tangenone, control, and with tangenone. These apoptotic figures here become much more frequent. So we see that this has an effect on human neutrophils. It has an effect on zebrafish apoptosis as well, which I'm not showing because the numbers are still very small. But what we found was that when we added tangenone, we saw a dramatic increase in reverse migration of neutrophils away from the wound. And we can quantify that here. This is the tangenone treated group, and this is the control now. And I just wanted to show this unpublished data that we did in collaboration with Daniel Irimer at Harvard. We used gradients here of leukotriene B4. This is maximal here at this point here, and the neutrophils are introduced on the right-hand side here. And what you would predict would happen is the neutrophils would move up to the site of maximum gradient here and stay at that point. And indeed, that does happen in many cases, but in both of the examples on this movie, and in a substantial minority of the, substantial minority of the conditions, these neutrophils will reverse migrate away from that high gradient. So under normal conditions, that happens about 30% of the time, with the other 70% migrating away. The other 70% staying at the site of injury, and this smaller fraction moving away. With the positive control, this is reversed. So lipoxin A4, this causes more reverse migration and more inflammation resolution. And our tangenone compounds here has a dose-dependent intermediate response, showing that as we increase the tangenone dose, we increase the reverse migration, and that's with human neutrophils. So we think that what we're showing here is that rather than just survival signals, things that neutrophils encounter at the inflammatory site act more as retention signals, keeping neutrophils both alive and keeping them present at the wound. And when these resolve, you either get apoptosis and clearance or reverse migration. And this seems to be co-regulated within the, at least within the zebrafish system. So for the last few minutes of the talk, I'm just going to talk about some work we've been doing with cystic fibrosis, or models of cystic fibrosis. So for those of you not familiar with the condition, it's a common genetic condition in Northern Europeans, and it is unfortunately fatal, and lung disease is the leading cause of death. The gene is the cystic fibrosis transmembrane conductance regulator, which is a chloride channel. And defects in this gene cause either a complete or partial loss of chloride flow at the cell surface. And the reason this causes lung disease is that chloride flux is important for regulating the amount of water in the airway mucus. So under normal circumstances, a thin, watery mucus is moved along by cilia from the bottom of the lung up to the trachea and into the throat, where we swallow, and any bacteria in this can then be killed by stomach acid. In the condition of cystic fibrosis, this mucus is dehydrated, it is sticky and is thick, it becomes infected, and the cilia are unable to clear the mucus out of the lungs, so the infected mucus then attracts neutrophils, which cause inflammation. They release nets to try and digest these bacteria, and those nets make the mucus even more thick and sticky. And actually, DNA's treatment to disrupt those neutrophil nets is a key part of therapy for cystic fibrosis. So this work was done by a really talented postdoctoral scientist, Audrey Bernoux, who came to my lab 4 or 5 years ago now to start doing some work on zebrafish inflammation in cystic fibrosis. She'd previously worked with Mycobacterium marinum, and she'd shown that, Mycobacterium abscessus. She'd shown Mycobacterium abscessus is a pathogen which is becoming increasingly important in cystic fibrosis. It infects about 25% of patients and is associated with a worse outcome from the disease. And it seems to be that people with cystic fibrosis have a specific defect in immunity to Mycobacterium abscessus. So, for example, the related pathogen Mycobacterium tuberculosis, they don't seem to have problems with, and in fact, there are anecdotal reports that they maybe have some protection against tuberculosis. What Audrey was able to show is that in morpholino, or complete genetic knockout of cystic fibrosis transmembrane conductance regulator, that there's an increased mortality to Mycobacterium abscessus. But interestingly, there's a relative protection against Mycobacterium marinum, which is a very close homologue of tuberculosis, again suggesting that there is some specificity in terms of the defect in immunity. And Audrey was able to show that that there is increased intracellular replication in the cystic fibrosis zebrafish, and that this is due to a reduction in the production of reactive oxygen species within neutrophils. So when Audrey came to the lab, she had shown that there was a primary defect in response to Mycobacterium abscessus in cystic fibrosis, which existed irrespective of defects in mucus and in host immunity. Which she was also wanted to test whether or not there were any primary defects in inflammation or in host tissue repair. So this cycle of infection, inflammation, and tissue damage, which is what causes the significant lung disease and cystic fibrosis, has always been thought to start here with infection. But we wanted to explore whether or not there were primary defects in these two aspects, which are very, very difficult to model in humans because of and indeed in mice, because of the chronic inflammation and damage caused by infection, whereas the zebrafish model doesn't have a problem with all that infection. So she used the transgenic zebrafish model that we've shown already, and what she was able to show is that following tissue injury, you had increased neutrophil recruitment to CFTR mutants and morphants, and this was quantitated here. So there's steeper recruitment here and slower resolution. And after some extensive investigation, Audrey was able to show that this was due to an enhanced reactive oxygen species production from the epithelium. So under normal circumstances, there's no reactive oxygen species produced at the wound, but following injury, there's a flash of cell rocks labeling here, which is enhanced in CFTR, both mutant and morphant. And again, this is quantitated here. And this is functionally important because if you block with DPI, which is a drug that blocks hydrogen peroxide, you see that that increase in inflammation with CFTR knockout is absent, and in fact the inflammation is suppressed in all of those cases. By adding back the hydrogen peroxide, we see that there is a restoration of the normal inflammatory response. But what I think is particularly interesting is that, as well as showing a defect now in inflammation, Audrey was also able to show a defect in tissue repair. So under normal circumstances, following injury, the tail fin will regrow. And if you knock out cystic fibrosis transmembrane conductance regulator, you see a reduction in the restoration of the normal tissue integrity. And this is shown in these two columns here. And again, this can be partially restored by inhibiting the reactive oxygen species or by removing neutrophils using this morpholino approach. So it looks like there's increased ROS production at the wound, and this is somehow responsible for increased neutrophil recruitment, and which is in turn responsible for tissue damage and impaired tissue repair. So Audrey wanted to test some of the treatments that we had developed in the lab to see whether or not these might be useful. So she used tansinone, identified as I've discussed already in this drug screen, and she showed that tansinone was able to restore the inflammatory response in CFTR knockout fish back down to baseline levels, suggesting that this rebalanced the immune response in cystic fibrosis zebrafish models. And just looking on the right here, she also showed that there was an increase in the tissue regeneration, again suggesting that this may have an effect on tissue repair. But many people, when we've been presenting this work, have asked, well, why is that the case? Why is there more ROS in the epithelium in cystic fibrosis? And the answer seems to be because of altered calcium flux. So this is a study of the reactive oxygen species, as we've shown previously. So this is the wild type, and this is the cystic fibrosis zebrafish. So you see an increase following injury in the control, but we were able to block this with two drugs that block calcium flux into the zebrafish. So this is quantitated here. So this is neutrophil number. This is reactive oxygen species, and you see no particularly significant change in the wild type, but in the CFTR knockouts, you see a reduction in the neutrophil number using these inhibitors of calcium flux. And we can also improve the tissue regeneration. So here using verapamil, I've shown most of this slide already, but you can see there's an improvement, which is modest but significant, in the cystic fibrosis fish if we use an inhibitor of calcium flux. And I should say this is a part of a class of drugs which are very, very widely used for the treatment of hypertension in humans. So they could be repurposed very easily. So very last couple of slides. One of the emerging problems in cystic fibrosis care at the moment is the emergence of colorectal cancer. So this is a common cancer anyway and causes a lot of cancer death in the UK. But people with cystic fibrosis have an unexplained 5-fold increase in gastrointestinal cancers. And if they are subject to lung transplants, this increases to a 30-fold increase, becoming really very significant for such patients. And interestingly, if you knock out CFTR in the intestines only in mice, these mice also have increased tumours, quite significant increases in the number of primary GI tumours in the mice. So we think that what might be happening is that this increased calcium flux that we've shown in the epithelium may be driving it, rises intracellular calcium. That is responsible for the activation of the Duox enzyme that produces hydrogen peroxide. So there's increase in reactive oxygen species production in the epithelium. So this is what we think causes the overactive neutrophilic inflammation, and neutrophilic inflammation can be a driver of tumorigenesis. But of course, reactive oxygen species also produce DNA damage, which can also contribute to the tumorigenesis. And at the same time, this intracellular calcium is a proliferative signal for epithelial cells, which is usually inhibited by calcineurin and NFAT. But of course, this is a target for many of the immunosuppressive drugs that patients with lung transplantation receive, suggesting again why these patients may have an increase in cancer. So we've recently been funded by the Cystic Fibrosis Trust to try and investigate this pathway using partly zebrafish models, but also human stem cells and a number of other systems. So hopefully we will be able to see whether or not this is true, and whether perhaps interfering upstream with calcium flux might prevent cancer in these patients. So that was us a few years ago, and this is us now. So I'd like to thank everybody in my lab, our collaborators, particularly Audrey for all of the work on cystic fibrosis, and Andres Floto, who's a CF physician in Cambridge. I've mentioned Daniel and Irma, and Phil Elks is still in Sheffield. He's a valued collaborator on a number of these projects. I'd like to thank all the funders, and thank you all for your very kind attention.
Well, thank you very much, Professor.
Hello, thank you, Dr. Steve. Wonderful talk.
It's very kind. It was my pleasure.
It was a really great talk, and we have some questions. So the first one is from Camila Morales from USP, University of São Paulo, and she said, "Great talk. Do you know what is the initial signal for the first neutrophil of the swarm to choose the place where it will release the nets?"
That is an excellent question. So we don't know that. So in the mouse model, it's obvious it's where you put the laser wound. In the zebrafish model, it's not so clear. It's always the same width, the same distance away from the wound, but it's sometimes in a different position along the tail fin. So the area where you see in the movies that I showed where it forms, that is one of the most common sites for neutrophils to migrate. It seems to be easier for them to get to that place, but the swarms can occur anywhere along the tail fin. So we have no idea why it chooses that spot, but it is a really, really good question. I think a very interesting area for research.
Well, we have another question now from Natalia Feitosa from UFRJ. Is there an increase of ROS in the lungs of the CFTR patients, as you see in the tail of the CFTR mutant fish?
Another great question. I would predict yes. I mean, the problem is it's very difficult to control that sort of experiment, because the lungs of those patients are very, very badly damaged. They are absolutely packed with neutrophils and inflammation. So and infection. So it's very, very difficult to find out whether there's a primary defect in ROS production in the epithelium, because it's very, very likely that any effect you see could be explained by the fact that there's just hundreds of neutrophils in there that are producing things, which is one of the reasons for using a model like the fish, where you don't have inflammation, you don't have infection. You can just take all those things out and look at the primary defect. We would like to do some work where we look perhaps at the bowels of patients with cystic fibrosis, because those the epithelium there, they also express CFTR, and the epithelial cells may have that same phenotype of the ROS, but they don't have all of the problems of the infection. So we're planning to do some studies on cystic fibrosis patients, but probably start with the bowel.
Thank you. There is also another question now from Veronica Lombardo from IBR in Argentina. She said, thanks for the excellent lecture. In the presence of A202, the number of neutrophils increase only in the wound area or in the entire embryo?
So I think the answer is just in the local area of the wound. So the hydrogen peroxide is a local chemoattractant for neutrophils. So I think it's a very, very primitive signal that that causes neutrophil recruitment. So when it's produced locally following tissue injury, it's one of the first things that recruits neutrophils to a site of damaged tissue. I haven't done the experiment where we add it to the whole fish and count, but I think it's predominantly a chemotactic effect for locally at the wound.
There is a question from Anta Krishna Tantri. I'm sorry if I don't speak correctly. Nice informative talk. What happens if you remove the pioneer neutrophils? Will the non-pioneer cells migrate or not towards the one site?
So, I mean, it's a really interesting question. I mean, it's also a theoretical question. I don't think in practice there's any way to do that, to remove that pioneer cell. I mean, we could try ablating it, but any ablation would cause tissue damage and cause potentially more recruitment there. So I think the neutrophils will still come. Not all fish have a swarm, but they all recruit neutrophils. So I think the neutrophils will still come. Whether they will still go to that same site and a second neutrophil would come along and take over that role because there's something special about that site, or whether it would just happen somewhere else or happen stochastically, I don't know. I think it's a question that we won't ever know the answer to.
I have a question on my own. It showed that there is some drugs that apparently has promising effects for cystic fibrosis patient, but fish at least. What's the next step to start treating patients?
So that is a good question. The treatment of cystic fibrosis, I didn't really go into it in the talk, but over the last year or so, it's been completely transformed by the release of new drugs that improve the trafficking of defective receptor to the cell surface and cause increased opening time of the receptor. And these drugs for many, many patients with cystic fibrosis have been completely transformative. So when we started the project, there was potentially more opportunity to treat than perhaps there is now. Those drugs don't work for everybody, so there are still a group of people that we could try them in. But the key things for things like tanchinones is to be absolutely sure that they don't have an immunosuppressive effect, because these diseases are driven by infection in the lung, and if we reduce the ability to fight infection, then potentially we might make things significantly worse. So there is a bit of background work to be done to understand whether or not they're safe to use in infection. I think the calcium antagonists potentially have more role there. These are drugs that are used routinely in hypertension, and they could be potentially repurposed. And if what we show is correct and is shown in mammalian or human systems, we might be able to show that that we can improve inflammation. We can also improve wound healing and tissue repair, and also prevent cancer, all with a single treatment. Now, I don't want to make claims that aren't true, but if all of these things are shown to be the case, then that would be a fantastic thing to be able to try in clinical practice.
Yeah, definitely. Well, we have another question now from Pierre-Olivier Angron. Amazing talk, thank you. On this side, do zebrafish mutants, CFTR mutants, develop cancers?
Great question. So we're studying that at the moment. We don't have aged fish that we've studied in to date, but we're going to look. And we're also knocking out things like P53 in the bowel as well, to see whether or not that can increase cancers in the CFTR.
Okay, I think you're ending our time as well. And thank you very much for the great talk and for the discussion. Thank you for the audience, for all the great questions. And I think you see you all in 2 hours. Yes, in 2 hours we'll come back with our last lecture of the day with Professor Marc Muller from Liège in Belgium. Thank you again, Professor Steve Ringshall, for this great talk.
Thank you for the great opportunity. It's been my pleasure.
See you at the afternoon.
Thank you. Bye.
Trabalhamos diariamente para criar soluções e equipamentos para a comunidade científica, contribuindo com o avanço da pesquisa biomédica latino-americana. Nossa missão é maior do que somente fornecer equipamentos. É proporcionar segurança com atendimento de qualidade e, principalmente, com muito respeito e atenção. Por isso, investimos em alta tecnologia e buscamos manter relacionamentos duradouros. Nosso compromisso é ser um parceiro confiável que compreenda as necessidades, a realidade e as condições de cada cliente para assim oferecer a melhor solução sempre. Entendemos os benefícios da pesquisa científica para a humanidade e isso nos estimula. Se hoje temos melhor qualidade de vida, maior longevidade, se vencemos um câncer ou fazemos uso de um remédio para dor de cabeça, é porque o avanço da pesquisa biomédica nos permite. Confiamos no trabalho dos pesquisadores, na ciência e na comunidade científica. E nos orgulhamos em fazer a nossa parte. Assim como você, somos apaixonados. Ciência é o que nos move, porque para a Alesco, pesquisa é para a vida.